Bismillahirrahmanirrahim. To dear colleague that was participating with us in the project of the microbiology of sinusitis during Hajj and in Makkah, first of all, I would like to thank you sincerely for participating in this. May Allah bless you and reward you, inshallah. I want to just demonstrate very briefly the practical steps of conducting the study. First of all, we have to identify the patient population. So please, if you have any patient who has the symptoms of acute rhinosinusitis, please include it in the study. And to define the acute rhinosinusitis, we have to have symptoms of 12 weeks or less of 2 or more symptoms of nasal blockage and nasal discharge, plus minus facial pain, plus minus reduction of smell. So if you do have a patient like this, please ask the patient to fill up this questionnaire. It's very brief, it's short, it's 1 page long, and we also have it in the electronic form. After filling it out, please collect the purulent secretion from the nasal cavity, from the middle meatus as demonstrated. And then please write down your name and signature and date in the 3rd page. One important thing is to label the secretions and the sample that you have accordingly to the patient database code that we have in the 1st page of the study and submit the questionnaire along with the sample to us. Thank you very much. Of the left nasal cavity, the tip is showing the inferior turbinate, and this is the middle turbinate. This is the uncinate process, and this is the bulla ethmoidalis. The aim of this study is to take culture from the middle meatus, which is the functional area right in here. So any purulent secretion coming from the middle meatus along down, even to the nasal floor, can be easily cultured using the culture swab, which is demonstrated right now. So the culture swab goes in, and we need to keep it where the pus is. For example, if it's in the middle meatus, for at least 10 seconds, so the purulent secretion will be embedded into the swab. Then easily we take the swab out and put it back into the culture media. So in order to collect the purulent secretion using the suction collector, you should avoid putting your finger on the control while getting in. You have to keep the shaft covered on the tip of the suction, and when you go into the purulent secretion, then you allow it to open so you avoid contamination of the nasal cavity. And then you take your endoscope, which is preferably a 0 degree, and then you put the scope on top, and then the suction comes from below. When we use the sinus collector to collect the purulent secretion from the nasal cavity, we have to make sure that the tip of the catheter is covered while we're going in. And once you get in, then you gently uncover this catheter so you keep it sterile and not contaminated with the nasal floor. Once you're in, you put your finger on the control, and then it starts to suction all the purulent secretion from the middle meatus. And then again, before coming out, please note that we have to cover the tip again and then gently come out so you avoid contamination to the anterior nasal. The other option is to collect the purulent secretion, if it's evident and obvious, by doing the manual nasal speculum, and then you introduce the swab inside, and once you get purulent secretion around it, you can turn your finger right and left and make it soaked and come out. If there's a lot of purulent secretion, this will be enough for the study. Please make sure and try not to touch or contaminate your samples with the anterior nasal floor while coming in and coming out.